Okay, so first, now that we're done with that, I'm first going to introduce our club president, Barb McPhee, so she can say a few words. Barb?
Thank you. Welcome to our big spring meeting. Usually we're in a big auditorium and lots of bee talk buzzing around, but this year it's very different. So we're dealing with it. We're certainly in for a great learning experience. For this meeting, we have co-sponsored Jeff Pettis talk with Hampden County. Please welcome members of the Hampden County Beekeeping Club. It's always good to cooperate with fellow beekeepers. I just want to remind WCBA members of our jar order. Go to our website, wcbamembers.wildapricot.com, and click on members only. There is a drop-down menu. Click store, and that will bring you to the jar order. Jars do not go bad, and our prices are very economical. Think positive. You'll need them come August. Also, at the same time, be sure to order the history of the Worcester County Beekeepers Association. Only $12 for a soft cover history book. We've already sold over 100 copies, so we're printing another 100 copies. Finally, we're putting together a calendar for 2022, asking photographers to send in pictures to me at barbshoneybees@gmail.com. We'll select a picture for each month and sell the calendars. Anyone whose picture is selected will get a free calendar. So start taking those photos. Now I would like to introduce Mark Lanczyk, President of Hampden County Beekeepers Association, to say a few words. Welcome, Mark.
Barbara, it's Mark Lanczakis. My email is kind of a truncated version of that, so it's Lanczakis, not a big deal. Good morning, everybody. I want to extend our welcome to the Hampden County folks that are online. I know we have a couple board members that are that I can see that are online, and I'd like to thank the Worcester County folks for the continued collaboration that we have with Hampden County. I think it elevates both of our organizations to share some resources and get involved with some of the topics that we're sharing on a regular basis. So with that, I'm gonna turn it over to Ann and Jeff, and we'll get the meeting started. Again, we're looking forward to a great discussion this morning. Appreciate you being here, Jeff, and looking forward to a great beekeeping season coming up this year after all we all been through.
Thanks, Mark. I'd now like to introduce Ken Washel so he can tell us a little bit about Jeff and introduce him.
Okay, I just, before I introduce Jeff, Barbara mentioned the history that I wrote of Worcester County beekeeping and some interesting stories. And just like we have the love for our bees in 1900, when the club was founded, they founded it for that reason, so they could attend meetings. But even when we founded the county association, it still involved traveling from Blackstone to a meeting in Barry, and the president, Mr. Fuller, had to take 3 trolley lines to get to Barry to the meeting, and then walk 2 miles to the meeting from where the trolley dropped them off, and then back again. So it was 2.5 hours to the meeting, 2.5 hours back by trolley lines. He missed the trolley line going back, kind of finishing up things at the meeting, and had to stay over at the Barry Hotel for $3 for the night, including his meal. So read some interesting stories, and there are all kind of documents in there going back to 1900, the founding charter. And so $12, and it's a deal. Get a warm spring day when the bees are flying in and out of the hive, put a chair right next to the hive. We can relax, watch the bees, and read through this history. You'll find it fascinating. You can buy it online at the store, or sending a check for $12 made out to WCBA to me, and that's in the newsletter. So that, I encourage everybody to buy this sold out first edition. We're on our second edition. And one other thing I just want to show, I've done some virtual meetings with students. Here's a, I don't know if you can see that, but a large bee. I sent to the school the bee outline, and then gave them a contest for a jar of honey. And this is a 6th grade rendition of a honeybee. You can see here. So it's pretty neat. And this was the winning entry, and she sent me the top 5 to display. So that might be something you might want to put in our calendar as well. Okay, with that, I'm going to introduce our speaker, Jeff Pettis, Dr. Jeff Pettis, who had served as the research leader at Beltsville Bee Lab for many years, became the president of Apimondia, the World Beekeeping Conference. And you all read, I'm sure, the detailed outline and hundreds of research articles. I've never seen any person with so many research articles with their name attached. And Dr. Callahan and myself both had the pleasure of working with Jeff for 4 years on a bee study here in Worcester County, and which had to do with the Asian longhorn beetle when it came in, to see the effects from the tree injection. So at this point, I'm going to introduce Jeff, and his first talk is going to be the queen and the colony. So with that, Jeff, it's all yours.
Okay, thanks, Ken. Well, thanks to Ken for the invite. Ken and I go way back, but and also Dick. I'll say a few more words about Dick and Ken and our interactions. But thanks to Barbara and Mark and Ann and the 2 associations for having me. We're going to spend some hours together. My first talk will be about queens. It'll start about queens, and then it'll go from queens to queen health to a bit about pesticides and some things about imidacloprid, some work we did in Worcester, Mass, and then some other things about pesticide exposure and how bees can fight that off, et cetera. So that'll be the first talk. And the second talk, I think, is a little more general. It's about world beekeeping. We'll take some travel logs around the globe. But it's a pleasure to be with you. I'm missing out on Leo's and having a nice Italian meal. I'm also missing out on sitting around during the break and having coffee. I'm seeing a lot of familiar faces. Barbara McPhee, I think I remember visiting your apiary when I was up there. I think that was one of the apiaries that we worked with. And before I go into my talk, the work I started doing on queens started with a conversation in the back room of your meeting hall in Worcester, Mass. Where's Dick? I'm looking for Dick. He's somewhere. Anyway, Dick, I don't know if you remember that, but Dick put this bug in my ear that shipping queens and queens in shipment might be subjected to high temperatures. And it took me a long time to act on it, but it was an idea that Dick Callahan gave me at a meeting in Worcester, Mass. I don't know how many years ago now. So thank you, Dick. And I will spell your name correctly in my next publication on that. I've got a few more coming out. But so I'm going to share my screen and start the talk. And it really is nice to be with you. Let's see. All right. Okay, so it says queen health and colony productivity, but the reality is that's only about half the talk, and the rest of the talk will be about some different aspects of pesticides, et cetera, et cetera. So it looks like everyone, you can see the screen and you can hear me. So let's proceed. Why are queens important? Well, if we ask beekeepers why they're losing colonies, you know, why did your colony die over winter, or why did it die in the summer? They often associate queen problems as being a major driver of why colonies are dying, the reason that colonies aren't doing well. And there's a couple of publications along the bottom there that you could look at that highlight this. But I'm going to show you some data from Canadian surveys, and we've done them in the US the same way. When we ask a beekeeper, why did you lose your colony? And queen losses, or poor queens, come out as often the number 1 or 2 reason that different beekeepers think that they lose colonies. So we know queens are important. They lay all the eggs in the colony, et cetera, et cetera. But even when we do these surveys, and this is in Canada, but the poor queens comes out as a major factor. We know that starvation, we know that varroa comes out. So like what Ken's doing right now, going around and looking at your winter losses, you need to be able to identify why your colony died. Now, the problem is, except for starvation and maybe a few other things, it's really hard sometimes to tell. But if you know good and well, if you go into fall with a poor queen, that colony is probably not going to come out the next spring and do well. So anyway, poor queens are an issue, and I'm going to talk about some things that could be involved in why queens are failing. So I'll first convince you that failing queens is an issue with some data from commercial bee colonies. Then I'll talk about temperature during queen shipments. And this was, like I said, dates back to a conversation I had with Dick Callahan at Worcester, Mass. And then I'll talk about a study I collaborated with in Canada, where we tested some queens that had been heat and cold stressed and how they performed in the colony. So I have 3 parts of this part, and then we'll get to more of the talk will be on later on pesticide effects. And this is just to highlight the last part of my talk is a collaboration between Dr. Steve Pernal and Dr. Martha Guarna at Ag Canada in Alberta. And they've been collaborating on many of these shipping studies with me. And we had an opportunity to do a colony level study in Canada a couple of years ago. The main way that we'll measure queen health in this case will be by her, by taking a queen. Here's your queen. We have to take a live queen and we open her up, open up her abdomen here. Inside there is a small thing. This is the tip of Sue Kobe. Many of you will know Sue Kobe, Sue Kobe's finger. This is a Spermatheca that has a lot of sperm in it. And this is a Spermatheca from a virgin queen, no sperm present. So you can take this Spermatheca out and look at it and go, ah, the queen's well mated. You can also open this up in a test tube. Oh, back up. You can open it up in a test tube and put 2 dyes in there, a green dye and a red dye, and tell what the proportion of live dead sperm. So you're looking at honeybee sperm. There are the tails coiled around. And this is the head of the sperm. If it's green, it's alive. And if it's red, it's dead. And of course, the male, the drones, mate with the queen early in her life, in her first 14 days of life, and she stores sperm. She stores all the sperm she needs for her whole life, 3 to 5 years, if they last that long, 3 to 5 years in the Spermatheca. So we're going to open this queen up, take the Spermatheca and look at the proportion of live dead sperm. And Anita Collins and colleagues at Beltsville developed this technique. So I'll be mostly reporting on percent sperm viability in these queens that we're going to follow. So when we heard beekeepers complaining about that their queens were failing and their colonies were failing, I asked them, can you send in live queens from failing colonies and from colonies in good health from the same apiary? So I got shipped a lot of queens from colonies that were failing, but no one would send me queens from colonies that the colony was in good health. So I went back and asked another time, and this was commercial beekeepers. And I said, can you please, within the same apiary, when you rate that colony, look at it. If the colony's failing, cage that queen and ship it to them. But then go to another colony that's performing well and send me that queen too. And I finally got a set of matched queens from the same apiary. The beekeeper said the colony was failing or the colony was in good health. And I'm going to show you that data on percent sperm viability from just those 2 types of queens, from an East Coast beekeeper and a West Coast beekeeper. So this is East Coast. I'm looking for my cursor. There's East Coast. This is the percent viability, which is just about 40% viability in the queens from failing colonies. The queens from healthy colonies, it was about 70. We'd like to see it up here. We'd like to see it about 85 to 90. But regardless, this is a reasonable viability. Both of these are reasonable. When it gets down to about 50% viability, we think that you start running into issues that the queen doesn't lay as well, there's poor brood pattern, et cetera. So we have an association. We know that the beekeeper rated these colonies as failing, and this represents about 10 or 12 queens, and this is 10 or 12 queens from the same apiary that were healthy, the same over here. So we have an association, but we don't know what's causing it. So that's what, then we begin to ask, could it be in the queen rearing or the mating procedure? Could it be there? Could it be something about shipping? This is my conversation with Dick Callahan. Or could it be where the queens are put? Maybe these queens are perfect. You're getting them from, I don't want to give shout outs to different queen breeders, but you get them from a real good queen breeder. They survive the shipping, but then you put them in lousy hive conditions. All of these things could probably affect how the queen performs. And I'm not going to spend time, but you know that poor weather, poor mating weather, and healthy drones are vital to, having good weather is vital to getting queens mated. I will spend some time on the shipping, and then I won't spend much time, but obviously the disease level and even pesticide levels in the colonies could affect queens. I point out there's a few more references down there if you want to go deeper into that. But we're going to focus on the shipping part, the fact that we've got good, well-mated queens here. We're going to trust them to the Postal Service and UPS, and they're going to come to us. So we're going to look at that part. So I went out to California, and I worked, in this case, it was California, not the Southeast. And I've got 6 different queen breeders. And I asked them to just take 20 queens and ship 10 UPS and 10 US Postal Service. So ship them to me in Beltsville. And I'm going to put thermocouples, small thermocouples you'll see in the next slide, and follow the temperature during that time. And then we're going to sacrifice the queens and look at the sperm viability. Because they came from the same group of queens from that queen breeder, I expect them to be identical. The thermocouples are these small little devices here. This is a nickel. That's a US nickel. There's the thermocouple. It's very small. You can put it inside the queen cage. You can put it beside the queen cage. It can go, and we can have it record up to every 10 minutes, every minute, whatever interval we choose to record temperature. So again, I've got 20 queens, 0.5% UPS and 0.5% US Postal Service. And I'm going to look at sperm viability. So that's what this figure is. These are the different breeders, and these were California breeders for this particular trial. And this is what we want. This is over 90% viability, and both US Postal Service and UPS both did a good job. There was no heat event or anything else, and high viability. Something happened here, and the UPS shipment, there's a difference in sperm viability. And so I'll tell you the rest of that story here in just a minute. What else do you notice in here? Well, look at this breeder number 4. Do you want to order queens from breeder number 4? You'd rather order from breeder number 2. I'll only say that queen breeders try and do a good job of breeding. They can't control the weather. And when I sampled this particular breeder 2 months later, their viability was up in the 80 or 90%. So this may have been things that were out of their control, local weather or whatever. So we're always trying to get above 80% viability. But back to the shipping thing. There was a cold event. These queens got chilled down to about between 45 and 50 degrees Fahrenheit. And that's what we're associating. That's the reason for the drop in sperm viability, was that these queens got cold during shipment, and probably cold in the airplane because it was summertime. So we have to assume it was because the plane, that the cargo hold was not thermoregulated well enough. But in any case, we do see a difference. But then I followed a lot more queen shipments over time. And I'll show you in the next figure. This is 15 different queen shipments. And we have to report in centigrade. So this is 40 is just over 100. And two of these shipments, the second and third shipment here, both went over 40. That's danger zone. Anything above about 39, 39 degrees C is in the danger zone. This one's getting close. But so these two were hot and these two other shipments were cold. These two shipments are cold. So it does happen. You get both queens that get heated and queens that get chilled. The problem is you can't tell when you get those queens, you can't tell that the queens have been heated or chilled, at least not now. We've talked about all kinds of ways of doing that, creating a shipping box that thermoregulates or putting temperature strips in there to know whether or not you get a heat event or not. And those could all happen. It'll be consumer demand that'll drive that. But right now, at least when I was following up, somewhere between 10 and 20% of the shipments were getting some kind of bad heat or cold event. So at least part of the queen problems could be related to that. But that's certainly not the only reason that queens fail. So we have this data from multiple shipments. Now I'm going to go back and say there's been work done on drones and drone exposure to pesticides, et cetera. We know that weather has a major effect. I published some things on the queen shipping back in 2016. This is about pesticides in queens and pesticide levels in the colony. So all of that comes into play. I don't want you to go away from this talk thinking, if I can just control the shipping, all my queen problems will go away. That's not the case. But I think we can do a better job in shipping. One way you can do that is just breed local queens, or go to your queen breeder and pick up queen cells and use queen cells to make nukes. So that's one way to get around it. So I'm going to conclude this part and then move on to the colony level study. We had an association that colonies that were performing poorly, the queens had low sperm viability. That was pretty obvious. We really need a lot of evidence to figure out, the role of other things like pesticides in the colony or drone quality have to do with our poor queen performance. So there's a lot of other things that go into producing a healthy queen that we don't want to blame it just on shipping. I've talked about pesticides, certainly in the colony and also in the queen ring process, queens can be affected by pesticide exposure. And there's this link between the colony performance and the viability within the queen. And now we're going to go and test that directly in Canada. We're going to take a group of queens, heat and cold stress them, and then follow their performance in the colony. So we're going to try to experimentally test this last idea that poor viability directly impacts colony productivity. This was my getting ahead of myself a little bit. There's at least one thing you can do to overcome a lot of queen issues, and that's breed local stock. I grew up with Georgia queen breeders, but if you have locally adapted stock that has gone through a couple of Worcester winters, then you've got a stock that is kind of geared towards that environment. So even if you have to import queens, if you can adapt and select for locally adapted stock, you're going to be further out ahead. And again, that was the idea that's the one thing you can do to overcome queen issues is just work with local stock. It's not always possible. We do need replacement queens. Okay. This is the study headed by Drs. Guarna and Steve Pernall at Ag Canada. We took 30 queens. We took 60 queens, actually 70 queens. We tested some of them to make sure they were viable. We tested a group to make sure that they had this fairly high viability at the beginning. I'm looking for my cursor. There it comes. So here's the high viability, anything over 80, 85% viability. So we test 10, we have 70 queens, we test 10 and sacrifice them and say, we've got decent viability. We take another group actually, before we introduce them in the colony, these have been heat and cold stress. So 42 and 40 for an hour. And look, the viability is down. So this represents this group here, the temperature stress. And this represents the viability of the controls. So we're going to just introduce 30 queens of each of these types into colonies and follow them over a season. And we use the same technique. This is all 100% alive, 100% dead, live and dead. And here we go. Is queen and colony performance affected? So we want to know, are the queens superseded? We want to know how well they do, and we're going to follow brood pattern, honey production, et cetera. So the first thing, we introduce the queens. The colonies are all started from packages, so they're all even. And the first thing we do is measure brood pattern. And we do that with this 100-cell grid thing. There's 4 missing cells in this one, maybe a 5th, but 4 missing cells out of 100. That's a good brood pattern. This is more than 20 cells missing. So we have a way of measuring. We measure 4 100-cell areas and get an idea, is the brood pattern good? And this is the data for brood pattern. So this is what you want. You want the blue. Over 90% of an area that she's laid in is constant, is sealed up. And then this is the one in the temperature stress. The brood patterns were spotty. Now this could be a sign of a good hygienic queen that's clearing out these few diseased cells. So brood pattern by itself is not everything, but it is an indication. We tend to look at it, and Ken looks at it for disease. He's going to look in these open cells. Is there any AFB or EFB or chalk brood? So brood pattern can tell you a lot. But first of all, the brood pattern in temperature stress colonies was poor. Adult bee population, this is sides covered by bees, sides covered by bees. So 20 sides, 10 frames completely covered. You're in the 20,000, maybe 25,000 bees in a colony, and this is half that. Of course, if they've got a poor brood pattern, they're probably producing a little less brood, worker brood. So you've got lower adult bee populations. What do you think that does to honey production? Well, you can imagine. Here's 60 kilos on average, kilos, not pounds. So like over 100 pounds of honey on average in these control colonies, and 2/3 less in the temperature stress. That could be due just to adult bee population or whatever, but regardless, you've got smaller colonies, weaker colonies, and they're not producing as much honey. That's very obvious. We went through the summer and we started with 30 in each group. We lost exactly 7. This never happened. We lost exactly 7 in both of the groups. So now we've got 23 good colonies going into the winter, 23 controls and 23 temperature stress. And we're going to ask, how do they overwinter? So the very last data, which is pretty striking, is that of these 23 in the fall, here's 23 and 23, look at my control survival. Very nice. I mean, yeah, that in a Canadian winter, that's very nice, 100% survival. Here, over 50% winter loss. Now, granted, these were already smaller colonies, but they were smaller colonies because we had heat and cold stress the queen. So this was a clear indication that this sperm viability matters, because that's the only thing we altered. We could have altered the queen in some way that we don't understand, but most likely it had to do with the reduced sperm viability, creating smaller colonies that then didn't winter as well, didn't produce as much honey. So we reduced the sperm viability. We saw poor brood patterns, lower adult bee population, much lower honey production, and the overwintering survival was pretty striking. So basically, we experimentally confirmed what we believed was already happening, that if you get a queen that has poor sperm viability, she's not going to perform well in your colony. And it could be due to shipping. It could be due to other things. It could be due to stress on the drones back when she was mated. So there's all kinds of things that come into play. And this is just an acknowledgement slide to thank Drs. Guarna and Purnell. You see pictured there. Project Apis M also provided funding for some of this. This is the Canadian Honey Council, Canadian Bee Research Fund. This is the Canadian National Bee Diagnostic Center, NBDC. Patricia Wolf did a lot of the analysis, myself, and then Dave Tarpey also at North Carolina State collaborated on this study. So a very nice study, and it's coming close to publication. We're always slow with publication, but this work is coming out in a publication very soon. So again, what's the one thing that you can do to overcome queen issues? Don't buy queens. No. I've already said it, breed or select from locally adapted stock. But again, that's not always possible. And I buy commercial queens and have them shipped to me all the time. You can ask for UPS or US Postal Service pickup at the branch. UPS doesn't like to do it, but you can request that, and that'll prevent it from going out in the UPS truck kind of thing. So there are ways that we can do that. Also, if you know your local UPS driver and they can get you those queens early in their round, that kind of thing simple things like that could help. So I think I'm shifting gears now. And now we're going to talk about pesticides, which is fine. We'll move into pesticides, and I'll get to the part that we did in Worcester as well as part of this. This is some collaboration with Dennis Van Engelsdorp and others, University of Maryland. And I put it in because I want to talk mostly about neonics, but we did sampling of bees on crop pollination. So the bees were on different crops that a beekeeper would pollinate, blueberries, cranberries, squash, things like that. And then we're going to look at the pesticides that are coming in on the pollen from those crops. So we had a field component and a lab component. The field component, we simply put pollen traps, picture their pollen traps on bees that were on these different crops for pollination. We collect the pollen, look at what plant type it is, and then we analyze for pesticides. And then the lab component, all we did was we take the same pollen, we feed it to newly emerged bees, we let them age for 12 days, we challenge them with nosema, we feed them nosema after we feed them the pollen, and look at how well nosema, this is nosema in the gut, look at how well the nosema replicates in those bees. So the crops, things like apple, blueberry, we did almonds, we didn't do the lab component of the almonds, and we do some cucurbits and melons. Anyway, so we collected from different crops around the country. And the first thing that was kind of interesting, the proportion of pollen from the different crops. Almond and apple, a high proportion, almost 100% of the pollen the bees are bringing in on almond is almond. The same for apple, it's very high. Look at these amounts of pollen from pumpkin or cranberry, et cetera. If you show this to a grower, they don't want to pay you to put bees on the crop. The deal here is that these are mostly New World crops that honeybees didn't adapt to, and they are transferring pollen. They're transferring it at a lower rate, and they're not collecting a lot of the crop pollen to pack it on their legs. So we know from a lot of different studies that, in fact, they do pollinate pumpkins. They pollinate watermelons. So it's not, but it's just they're not collecting a lot of excess watermelon or pumpkin pollen. Whereas in these two crops, they are. The vast majority are these. So now I'm going to analyze the pollen, the total pollen that was collected. So any of the pesticides that we see in these crops are going to be not from the crop itself, like over here with apple and almond, but from surrounding vegetation. So the vast majority in these others are, the pesticides are going to come not from the crop, but from surrounding flowering plants. This is just a bee on an almond flower. Again, high proportion of almond pollen collected and packed on the corbicula. This is where Ken says to sit back and read your Worcester County beekeeper history and watch the bees coming in. And my bees are now collecting red maple pollen, which I'm very pleased to see. This is the number of pesticides detected. Just the actual number is the white bar. And the dark bar is an estimate of the total pesticide load. So let's just take apple. Over 14 pesticides, 14 different pesticides, and the amount is moderate. So there's some exposure, moderate. Blueberry, fewer pesticides and less amount, whereas pumpkin, we get the highest total amount of pesticides, but only about 6 different pesticides. What do you make of that? Well, what I want to point out, so we have all these different crops across the country, and there's been a lot of concern about neonics. Let me show you, if you break this pesticides down by class, by fungicide, neonics, et cetera, and you'll see that, you'll see what type of pesticide the bees are exposed to. And this is what this is supposed to be about, that we get a lot of attention on neonics because they're systemic, and they should get a lot of attention. But fungicide exposure is also of concern. So this is by the number of chemicals present by the different class. So this is fungicides. Here's our neonics. And because we'll start with the neonics. From this bar to this bar, where did we detect neonics? Only in apple. So that's apple is the first bar. Look at the fungicides. We detected fungicide exposure in every crop we looked at. So high fungicide exposure, high pyrethroid exposure, fluvalinate and other things are pyrethroids, and then some organophosphate exposure. So I point this out just to say that this was directly from the pollen that we collected off the crop or around the crop. And in this particular case, for most of these crops, the neonics were not as well represented as we might think, but the fungicides were. So now we'll do the last part. We'll do the lab part, which is the last part. And to do that, let me back up. We're going to feed this pollen, and it has all these different mixtures of pesticides in it. How are we going to make any sense of it? Well, the only way we're going to make sense of it is by taking an epidemiological approach and saying, if we see these 3 compounds, whatever they are, present, do we see an increase in nosema levels? Or what are the combinations of pesticides that cause increase in nosema levels in this case? And it goes to human medicine. If you want to know about lung cancer, you look at potential causative factors of lung cancer. So we know quite clearly from the tobacco industry that smoking doesn't cause lung cancer, right? There's no cause and effect. But if you smoke, you're much more likely to come down with lung cancer. So the relative risk, this RR, relative risk of getting lung cancer is much, much higher if you're a smoker. And the same for coronary disease. Even a 3 or 4-fold increase in relative risk is high. So smoking, your relative risk of coronary disease goes up, but lung cancer is off the charts. So we're going to do this for bees and nosema. And I'll show you just a little bit of data. This is a fungicide, chlorthalonil. It's widely used. This is another fungicide, and this is one that there wasn't much of an association. So this is almost 3, and this is 2.5 or so. This is enough of a relationship to say that when these 2 products are present, we got increased nosema. For true pesticides, not fungicides, this one, there was an association by bifenthrin. Fluvalinate, which we use to control varroa, there was an association. Nosema levels went up. But imidacloprid, there wasn't an association. So again, it's trying to make sense of real world situations of exposure and what risk it might pose for bees. And I'm not drawing any outstanding conclusions from this. The one conclusion I am drawing is that we shouldn't overlook fungicides. Fungicide exposure can cause problems in bee health. And that's in this paper that we published. But there's always a bright side. You got to have a bright side. This is a different paper. And this is Dennis Van Ingelsdorp and I and others. And we talked about entombed pollen. These dark cells here, and you can look for this in your own colonies. When bees find something they don't like, a dead rat, or in this case, pollen cells that they don't like, they cover it up with propolis. So here they're covering this pollen up with propolis, and this pollen they're leaving alone. So this pollen is not covered up, and there's an entombed pollen cell, and here's some other ones. When we analyze the pollen here versus here in the same comb, we came up with some interesting fungicide exposure. So here's the control pollen, unsealed, low levels of fluvalinate, coumaphos, and chlorthalonil, and chlorpyrifos is the ag chemical. The entombed pollen, high fluvalinate, high coumaphos, and really high, I'm covered up on my screen, but very high levels of chlorthalonil. So the chlorthalonil levels were very high compared to the same pollen or the same comb, pollen from the same comb. What we think is happening is this chlorthalonil is changing this pollen, changing the degradation of the maturation of the pollen. It's pulling the fluvalinate and coumaphos out of the beeswax. So the fluvalinate and coumaphos should be in this beeswax because it sticks in the beeswax. But the fungicide we're proposing changed the way this bee bread was maturing, and it pulled these other two compounds out. And in fact, there's high chlorthalonil levels in this entombed pollen, and the bees will tend to ignore it. So we associated entombed pollen with a slightly increased risk of the colony dying later on. Not a real cause and effect, but if you see a lot of entombed pollen, you might be subjected to pesticide or fungicide exposure. And you can go to that paper, 2009 Journal of Invertebrate Pathology, and read more about it if you like. But the good news about this is that the bees recognize it and they seal it up. So the bees are sealing this up, and they won't touch that pollen. They ignore it. So they're isolating it. So that's the good news. That's the really good news. Okay. Bees are exposed to multiple pesticides in crops, and fungicides and pathogens interactions are common. We see that repeatedly, that fungicides should not be ignored. Okay, a little closer to home. We got commissioned with APHIS, Animal Plant Health Inspection Service. I mean, you guys have lived through this Asian longhorn beetle control program. So Worcester, Mass had an active control program going on, and New York City, parts of New York City, Brooklyn, and Queens had an active program going on. We got a grant from APHIS to look at were the treatments that they were doing trunk and soil injection. Now, I'll talk a lot about trunk and soil injection of imidacloprid in red maple. Was that exposing our bees to harmful levels of imidacloprid, and could it affect bee health? Well, you're about to see red maple bloom, right? And it is an important spring buildup. So it was a really good question to ask. And thanks to the help of Ken Wurchel, Dick Callahan, and a lot of others, we were able to address this. So I'll go through that. This is Jody Johnson, the PhD student who's working. We worked in Queens, primarily in Queens. We worked in cemeteries where we had mature trees, red maple trees. And we looked at trunk injection, and then the other treatment is where they inject the soil and have the roots pick up the imidacloprid. And APHIS had already worked out the details of the dose, so we just worked with what they were doing, whether they were soil injecting or trunk injecting these trees. Red maple is very interesting. You can have a tree that's nothing but female flowers, and you can have trees that are nothing but male flowers. These are, you can see the pollen here coming out on these male flowers, and you can have trees that are mixed. In general, you tend to have a female tree or a male tree, but you can have trees that are mixed too. And we tended to work only with either male or female, just because we wanted to see if there was differences in expression. But we know red maple is an important buildup crop, and that's what this shows. This is from Worcester, weeks, and I don't tell you the exact dates, it's not important, but we started trapping and we got no pollen yet from red maple. By week 2 into that, we're at about, what is that, 13% of the trap pollen was maple, red maple. Then you're up at 27%, almost 50%. By week 4, we're changing these pollen traps every week. By week 4, we had 45, 40, 50, almost 50% of our pollen coming in during that time was red maple. And if you collect this over this time period, this 2.5, 3-week time period, that's a high proportion of your spring pollen coming from red maple. The other are things like willow. So if you took the rest of this bar and went to 100%, it'd be willow and other things. And if they're injecting the trees or treating the soil, how much imidacloprid is in this red maple? That was the question we were asked to address. So here's some data on that. 3-year trunk injection, and this is in pollen samples, it was undetectable in trunk injection. Here as well, in trunk injection, it was undetectable. 1-year soil versus 3-year soil. Soil injection, it was very clear, caused higher levels in pollen. So we had some nectar data. The nectar, it doesn't come out much in nectar. There's almost no detection in nectar. But in pollen, if you want to, the safest means of treating the trees would be trunk injection. And we gave this information to APHIS, and they're continuing to do control programs in different places. I know there's one in Pennsylvania going on now. So if they want to protect pollinators, you stay away from soil injection. But even with that, supposedly this is 10 parts per billion. Supposedly 20 parts per billion is where you start seeing effects on bees. So even these levels, while they're not good, I won't say they're good, they're below what we believe are levels that would have a major effect on the colony. So we did not see, in all the work we did not see effects on the nuke boxes that we put there and things, but we didn't have time to do the long-term studies. But these are low levels, but there is some accumulation in the soil and it gets expressed in the pollen. And I would say this 10 parts per billion is half of what we think would have an effect on the colony or have effect on bee behavior. So it's not benign, but it's not a great cause of concern. I can say that some of the ornamentals that the Home Depot and others are treating, I'd be much more worried because they're treating the soil in those potted plants, and those potted plants are blooming in your yard. And yeah, there can be a lot of direct exposure, but at least so far, we gave this information to APHIS, and I would say that by and large, trunk injection looked very safe, soil injection less so, but they can decide what process they take. And so they've used this information in their control strategies. Oh, back up. So the take-home message here was that soil treatment yielded higher residues in pollen versus trunk injection. Trunk injection, we did see differences between male and female flowers, but it wasn't that interesting. But we did see some difference between male and female, how much imidacloprid was in the male and female flowers. And this is this no observable effect limit of 20 parts per billion. And that the highest we saw was 10 parts per billion. And this is Robin Rose, who helped fund the study from APHIS, Jody, and Dick, who I thank for his help in carrying it out, also for his wisdom on queens. And Fuzzy Ken, this is the only photo I had of you, so my apologies. But you have a valuable resource in Ken, and so even though he retires next year, keep tapping into his expertise. Ken was invaluable in helping us carry out these studies. With that, I'm going to close this part of my presentation, but I will invite you. It's UFA, Russia, Apimundia, September. Sometime in probably September, we don't have the exact dates, of 2022. We postponed it by a year. And I'm going to be around for questions now, and then we'll take a break, and then we'll be back with more. So, but thank you for your attention on this. And again, don't go away thinking that all your queens are bad, and don't always blame the queen breeder. They're doing a good job to try to rear good quality queens. So with that, I'll stop screen sharing, and we'll open it up. Thank you.
If you have a question, please push the raise hand button. Please don't type it into the chat. Just push the raise hand and I will call on you. So the first one with a question is Mark. So if you want to unmute Mark and ask your question.
Yes, thank you. Thank you, Jeff, for a very informative discussion. A question with regard to sperm viability. I mean, obviously, it's very clear if a sperm is dead or alive.
Mhm.
Is the bad brood pattern because she's trying to fertilize an egg with a dead sperm, or is there some genetic change or the genetic quality of the sperm that are alive that are causing it? Is it just dead or alive, or is there some genetic modification that's going on through that stressing process?
That's an excellent question, and I don't have an answer. No, I wish I did. So the only way I could address it is that Anita Collins did something where she took and did artificial insemination, and she did 25% dead, 75% alive, and she did 50-50, and she did 75% dead and 25% alive. And then she watched those colonies build up. And at 50-50, she was starting to get spotty brood pattern. And at the 75% dead, she got lousy brood pattern. So I think it's that she's trying to lay, she's thinking she's fertilizing, but half the sperm, they're just not being fertilized. And so she might be laying a drone egg and they're clearing it out. But you're right, it could be, Mark, that that the sperm is actually alive, but it's compromised. And we just haven't gotten that far in the understanding of it. So I don't know the answer to that.
No, understand. Thank you.
Okay, Skip, you're next, and then Dave and Marcy will be after Skip.
Thank you. I'm assuming that the queen has to be sacrificed in order to make those sperm studies. Is that true?
That is true, Skip. Yeah.
So the question becomes, are there any technologies on the horizon or that may exist that allow you to do those results of the studies on the sperm while the queen is alive?
So the best I can think of, and I've explored it a little bit with nosema and viruses in queens, is you can take a queen and you put her in a small dish, like just take her off the comb, put her in a small dish for a minute, and she will usually defecate. She'll usually poop. And you can take that sample and look for different signals in her feces. But beyond that, I don't know of any, like a full body scan, like we could put them in a little. No, but I do think it's along that same line. This is just a general, I'm getting off subject here, but you have summer bees and bees that go into winter, we call winter bees. And I think we're going to get to a point where you can take a small sample of your bees, crush them up, and look and see how overall healthy they are, the workers, in the summer and also the fall. You'll be able to tell stuff like how much nosema is there, how much they've been parasitized by varroa and stuff like that. And yeah, the queen special, I don't know if people are going to take the time to develop some techniques with just the queen herself. But the one thing I thought of was looking at her feces, you can at least get a health report. Yeah.
So no MRI machines yet for that.
Not that I know of. Yeah.
Thank you.
Okay, Dave, you're next, and then Becca after you.
Hey, thanks, Jeff. That was excellent talk. It was really interesting. We're located kind of on the edge of the longhorn beetle zone, and I was interested in understanding more about the 3 and 1 year time points, and also if there's any sort of indication as to if the maple trees in our area were injected, how long does that injection remain in the tree, and how long is that? Is there any indication as to how long that pesticide remains in the pollen for the bees?
Perfect question, Dave. So when not in Worcester, but in Queens, we had, so they were trunk injecting it, if I'm remembering correctly, they were trunk injecting 1 year, 2 years, 3 years. They actually went back to the tree and re-injected. So it was just a question of logistics and stuff, whether they used trunk or soil injection. There was arguments for both. I can't remember which they said was more expensive, but regardless, they were going back in both cases and injecting the first year, second year, third year. So in Queens, we had the chance to start in the first year with third year soil and tree injections, and we followed them for 3 more years. So we've had some 1 year, 2 year, and 3 year sites, and we were able to follow them for 3 more years. So now we actually get 6 years of data in only 3 years. And the good news was in both the soil and the trunk, it started to turn down. It didn't go away, but after like at least stayed high from the 3rd year to the 4th year, but by the 5th and the 6th year in both the leaves and the pollen, the amount was starting to go down. So that, I mean, that's good news. It's not going to stay around forever. I guess that's about the amount. And we need to get this publication out. We gave it to APHIS, but Jody, my PhD student, is like, Jody, you know, there's work going on right now in Pennsylvania. And they were like, there's almost nothing known about movement in hardwood trees. And like, well, we do know quite a bit. We just need to get it out there. And some of those questions are answered in the more details about 1st versus 6th year levels. But the good news was it did begin to turn down.
Great. Thank you so much.
Yep.
Okay, G. Moraes, you're up next for a question.
Hello, I have a question about temperature effect on the queen during the normal environmental conditions, like a very cold winter or a very hot summer. How does that impact? Has anyone done research on that?
Well, the cold is a little easier to deal with. So even in the winter, she's going to be in the middle of the cluster. She's not one that's going to cycle out to the edge of the cluster. So she's going to be protected. They're going to keep that cluster temperature warm enough that I'm not worried about the winter situation. I did some work in Southern California, and we put thermocouples all the way across the brood nest, all across all 10 frames. And it got to 45 degrees C, which is really hot, like 120. I mean, it just, and the outside couple frames got above like 38. So they still were able to thermoregulate. They bring water in, they cool it as best they can. But the outside frames got a little hot. And I think the drones that are out there might be getting cooked. So the drones might be susceptible. I think the queen, almost always, they're going to protect that brood nest and cool that brood nest. So I don't think there's anything to worry about extremely hot weather, except for the drones. I think the queen is usually in the brood nest area and she's pretty well protected. But I do worry about drones. I worry about drones.
Thank you.
Any other questions? I don't see anybody else with a raised hand. So I guess that might be it for questions, Jeff. So why don't we break for 15 minutes? It's just 10 o'clock, and I will meet everybody back here at 10:15 for Jeff's next talk.
Sounds good. Thanks, Ann. We'll be right back after these messages.